Good morning, class. During this short video, I will discuss Henrietta Lacks and her immortal cells, also known as HeLa cells. Henrietta Lacks cells were established in 1951 and were the first cells that were cultured in a laboratory. These cells were cancer cells that multiplied and grew at an increased rate as well as infected many other cells. These cells are still used today in numerous labs across the world and have played a vital role in medical research, creation of vaccines, and the knowledge of cells in general. Although most people have never heard of them, these cells have been a major part of the history and development of scientific research in the medical field. I will explore how the cells are retained, how they have been divided and multiplied, how they have had an effect in research and other areas, and how the family of Henrietta Lacks has been involved. Henrietta Lacks was born in 1920 in Roanoke, Virginia. She was married to her cousin David Lacks, with whom she had 5 children. In January of 1951, she went to Johns Hopkins because she had been feeling a knot in her womb. This was the only hospital in the area that would treat black patients at that time. The doctor that she was seeing took a biopsy of the mass that was on her cervix and determined that she had a form of cervical cancer. During her treatment, samples of Henrietta's cells from her cervix were taken without her knowledge or her permission, with one of the samples being cancerous and the other being a sample of healthy tissue. During one of her treatments, she asked to be admitted because of severe abdominal pain that she was experiencing. She received a blood transfusion but remained in the hospital for a few months before her death on October 4th of 1951. This timeline shows the impacts of HeLa cells and the scientific advances that have been made possible because of them. These cells were infected with everything from mumps to measles to herpes, which led to the modern field of virology. HeLa cells were susceptible to polio and were used in the largest vaccine field trial to date. They also helped prove that HPV causes cancer, as well as helped in HIV research, which helped identify a key receptor of the infection mechanism. These cells were useful in advancing the use of lab materials by being used to develop processes for growing cells in bulk and how to melt them without killing them, testing glass that is used in labs, and research on nanoparticles. When it comes to genetics, HeLa cells were used when it was discovered which stain made chromosomes visible, which led to the discovery of what genetics link to different diseases like Down syndrome, which has helped in genetic disease diagnosis and testing. These cells were fused with mouse cells, which was the first cross-species hybrid, which helped in mapping human gene, blood type identification, and cancer drug development. The hardiness of these cells has allowed scientists to devise a method for isolating a single cell long enough for it to replicate itself perfectly, which plays a role in cloning, gene therapy, in vitro fertilization, and stem cell isolation. HeLa cells were sent into space by NASA before any astronaut to see how gravity affected human space. They've also been used in studying the effects of deep-sea pressure, bacterium research on how tuberculosis and salmonella affect human cells, testing of cosmetics and pharmaceuticals replacing animals, and the development for treatments of diseases such as AIDS, Parkinson's, influenza, leukemia, and hemophilia. As you can see, HeLa cells have had a major impact on a variety of scientific endeavors and numerous areas of research. Today, these cells are being replicated all over the world and continue to pave the way for new discoveries. As mentioned before, Henrietta's cells were taken without her knowledge or permission. At that time, permission was not required, so it was typically not sought. However, when information about the cells started to surface, members of the press began to question who the cells came from. Doctors attempted to deceive the media by giving names such as Helen Lane or Helen Larson. Henrietta's real name was not released until the 1970s, almost 20 years after her death. Rebecca Skloot, the author of The Immortal Life of Henrietta Lacks, was a student who became obsessed with Henrietta's story, and she was determined to learn more about her and her family. A contamination issue in a lab where the cells were being used called for a scientist to track down members of the Lacks family for more samples so they could attempt to fix the contamination problem. From then on, the family became a part of the research. Deborah Lacks, who was instantly drawn to the research because she wanted to know more about her mother who died when she was an infant. However, Henrietta's sons became angry when they learned of the monetary value of their mother's cells because they felt entitled to some part of that. These cells were the start of a multi-billion-dollar industry. There was an agreement made with the family that scientists had to get permission to use HeLa cells, as well as any research published had to include recognition and thanks to the family. This agreement did not include any type of compensation. Recently, her eldest son, Lawrence Lacks, who is the executive of Henrietta's estate, is still seeking compensation from Johns Hopkins University and has recent plans to file a lawsuit. Henrietta's cells have been considered the foundation of modern medical science, which I believe has been made very clear. They have been in the forefront of medical research and scientific developments. The immortal life of Henrietta Lacks has been turned into an HBO movie starring Oprah Winfrey as Deborah Lacks. This movie is set to debut on April 22nd, and it will finally give Henrietta the recognition that she deserves.